Hello students, welcome to Misostudy again. So as we were discussing about the chapter strategies for enhancement of food production, in the today's session I will be talking about some questions which are related to your CBSE. So I have included around 10 questions and we will be talking about those 10 questions, some questions 3 marks, 2 marks and some 1 marks and some are a very long answer type question. I will tell you how to attempt such type of questions, and also these are the questions which we have collected, and some are from the previous year exams, previous year CBSE exam. So let's start the today's session. So first 3 questions are of 1 mark question, right? So first question indicate, mention the role of a genetic mother in MOET. Now what is MOET? MOET is multiple ovulation and embryo transfer. Right. This is called as a MOET. Now to talk more about this procedure of MOET, in this what was happening, FSH was given. What is FSH? That is a follicular stimulating hormone is given to a superior female, and superior female is that female which can give rise to a large number of or large quantity of milk, and the quality of that milk is also good. So that is called as a female. So egg is taken from that female after their treatment with the FSH, which is a follicular stimulating hormone. That act as a genetic donor, or that as a donor of this, and later on after the fertilization with a specific sperm from elite bull, from you can say superior bull, which can give rise to a superior kind of offspring. After that the fertilization happen, 8 to 32 cell stage, they are implanted into a mother, into a surrogate mother. And basically here role of genetic mother is to give egg after super ovulation. After super ovulation because of treatment with follicular stimulating hormone. Now a word is written over here, that is a genetic mother. Genetic mother is different from a surrogate mother. Surrogate mother is that mother which carries the baby for a 9 month. When we talk about a human being, whereas a genetic mother is that mother which is the actual donor of one of the gamete. So in this case only that mother act as a donor, so they gives egg. So this is a role of a genetic mother. Genetic mother doesn't carry baby for the whole process of development of baby, doesn't carry that, they just give a egg. So that is called as a genetic mother. So hope the first question is clear to you. Let's move to the next question. Now once again the next question is of 1 mark. Let's see. Mention the economic value of Apis indica. In the sessions we have discussed about apiculture. Apiculture is also called as a bee culture. Apiculture is also called bee culture. Why bee culturing is done, and mainly the Apis indica is done. This is done for 2 things. The first one is for honey. Honey has antibacterial properties, whereas another, their culturing is done for another thing which is called as a wax. And wax is used for the preparation of cosmetics. For the preparation of cosmetics. Right? This is the economic value of Apis indica, and mainly they are cultured for wax. Mainly they are cultured for wax. Hope you know now, you won't do any mistake if such type of question come regarding the Apis indica. Now let's move on to the next question. So, next question is related to name any disease the Himgiri variety of wheat is resistant to. So, Himgiri variety of wheat is prepared with the help of which strategy? That is a plant breeding. Plant breeding. And this particular variety is resistant to two diseases. What is the name of two diseases? The first name is the leaf rust. Second is called as a stripe rust. Leaf rust and stripe rust, these are the two diseases from which the Himgiri variety is resistant to. We have studied the same in the session, so hope you remember the same. Now, let's move on to the next question. So, this is a 2 marks question. This is also related to MOET, multiple ovulation embryo transfer. Study the flow chart given below and first identify the event that take place at stage A and B respectively. State the importance of technology explained above. Now, a cow is administered with FSH. Now, what is FSH? This is a follicular stimulating hormone. Now, this particular hormone, they help in which process? That is for multiple ovulation or a super ovulation, and that is being administered to a female mother, so that a genetic mother, like we have discussed, and when it is administered to a female, the super ovulation happens in them. So, the A which is written over here, this is called as a super ovulation. That means here the first question indicates that we have to explain about the process which is happening. So, the first process which is happening, that is called as a super ovulation. 6 to 8 eggs per cycle they are derived. Normally only one egg come, but in this case 6 to 8 eggs they come in each cycle, artificially inseminated. So, what is happening now? Fertilization. Are you getting what I am trying to say? Artificial insemination means artificial introduction of a male gamete. Fertilized egg at 8 to 32 cells are recovered, and after that is implanted into surrogate mother. So, or implantation or implanted to, in surrogate mother. Surrogate mother is another mother, like I have discussed. Here the super ovulation, this type of cow where the FSH is introduced, this is called as a genetic mother. Because genetic mother is a donor of egg, whereas the another mother which carries that baby for a period, for a longer period, that is called as a surrogate mother. Now, we have identified the A and B. Now, I have to state the importance of the technology explained above. The above technology is called as a MOET. Like I have discussed also, that is a multiple ovulation and embryo transfer. Multiple ovulation and embryo transfer. Now, we have to identify the importance. In this case, what we can do, we can produce a superior female or superior male, and female is produced. right? Those females which are produced, they can give large and large amount of milk. So, on lactation, such type of cows produce more milk, right? Whereas the male is that which can produce superior next generation or superior zygote they produce. Basically, the involvement is that we have to improve the herd size. So, one of the importance is improvement of herd size. Right. So, this was a question number 4 which account for only 2 marks. So, this is how you have to attempt. Now, let us move to the next question, question number 5. Very easy question, very direct question it is, a 5 marks question, sorry 2 marks question, question number 5. In an agricultural field, there is a prevalence of a following microorganisms and the crop diseases which are affecting the crop yield badly. So, which type of crop we should grow? First is a white rust, second is a leaf and the stripe rust, black rot and jussets are there. So, we have to look for those varieties which are resistant to different type of pest. To talk about a white rust, we have developed one variety which is of a Pusa swirnim. Pusa swirnim. Right. Second, it is written the leaf and the stripe rust disease. In the first 1 mark question we have discussed, this is for the variety which is available, that is called as a Himgiri. Himgiri. Now, let us talk about a third which is a black rot, which is a Pusa shubhra. Right. Here the last one is a jussets. Jussets, for we have a variety which is a Pusa sem 2. Pusa sem 2. These are the different varieties which is available. First is a Pusa swirnim, second is a Himgiri, third is a Pusa shubhra, and fourth one is a Pusa sem 2. So, these are the different varieties which is available, and these are produced with the help of which procedure? Plant breeding. And these varieties, they are pest resistant. They are pest resistant, right? Now, let us move on to the next question, question number 6. Enumerates 4 objectives for the improving the nutritional quality of different crop for the health benefits of human population by the process of biofortification. So, biofortification is that process in which the overall the nutritional quality of a crop is increased. So, what we want is that we should get enough amount of protein, we should get enough amount of vitamins as well, the oils as well, the micronutrients as well. Our major focus is that. So, we have to define the 4 objectives. 4 objectives, a very direct objective, direct question is that to increase quality and quantity of protein, quantity of proteins, right. Second point is to increase quality and quantity of oil. of oil, and the third one, to increase vitamin content. Vitamin content, right? And the fourth one is to increase micronutrient and mineral content, because whenever this content is high, because all of these, they are important for a proper growth. When this content of different substances, like we have discussed, the first, the protein is there, second we have discussed the oil, the vitamins also, and the mineral content and the micronutrient, all of these, they are important for a sustainable growth, for a proper growth of body as well as brain. So these are the four objectives, that's why we go for a biofortification. Today we have the biofortified rice, today we have the biofortified corn also present with us. Now let's move on to the next question. Question number 7. This is of a 3 marks. Enlist the steps involved in the inbreeding of cattle, suggest 2 disadvantages of this practice, right? Now we have to find out the process of inbreeding. So question arises, what is a inbreeding? Inbreeding is a process by which a mating happened between 2 individuals which are related to each other, or you can say which are having a common ancestor, or they are related to each other, and they are, you can say, they are belong to a same breed. So mating between 2 individuals, right? First, they should be related. Second, they should have a common ancestor. They should have a common ancestor. Also, they should have a common breed as well. This is called as a inbreeding. We have discussed about the breeding. This is called as a first inbreeding. Now we have to enlist the steps which is involved in the breeding of cattle. Now first step, come to this side. The first step for the process of inbreeding is finding the superior male and superior female. That is, first of all, that will look for a superior male and superior female. And superior female. Superior male and the superior female. So superior male will act as a father, whereas a superior female will act as a mother. Right? And one thing should be clear in our mind, that they should belong to a common breed, they should have a common ancestor, and they should be related to each other. Now after that, they will be allowed to hybridize, or they will be allowed to mate. Mating between parents. Right? After that, evaluation of progeny for desired trait. Right? Now in this case, whatever progeny which we are getting, they will be allowed to undergo the mating for 4 to 6 generation. For 4 to 6 generation, they will be allowed to do that. Right? Now what are the benefit of these? So that whatever the dominant or positive alleles are there, that should get accumulated. whatever those recessive or you can say the negative alleles which are there which may cause disease, they should be removed out by choosing or by the process of evaluation, right. So, after 4 to 6 generation, we will check them for 4 to 6 generation and later on they will be used. Now, one more thing is that when we talk about a superior male is that male which can give rise to a superior offspring, whose offsprings are all healthy. Whereas, the superior female is that female which can give rise to a large amount of milk while lactation. Suggest two disadvantages of this practice. The major disadvantage is that there is a loss in productivity and fertility after 4 to 6 inbreeding, right. This is the major consequence. Another consequence that you all must have heard, that is inbreeding depression. We have discussed in sessions. These are the two different things which you have to explain, right, inbreeding depression and also decrease in productivity and fertility after 4 to 6 times of inbreeding. Now come back to the question. So this is how you have to attempt these 3 marks question. Now let's move to a next question, the last question which is of 3 marks. After that we have is a 5 mark question. Now, what is a blue revolution and name to fresh water and to marine edible fishes? So, what is a blue revolution? So, people who are living in a coastal areas, they are depending upon these fishes, basically they are depending upon the fisheries for their breads even. Even when we talk about the total content of the micronutrient, total content of different fats and the lipids which is present in them, they are very high. So, that is why a revolution is there towards the breeding methods in case, and that leads to a blue revolution. That means to increase in the fisheries industry, because the fishes are a rich source of the lipids, a good lipid are there, omega-3 fatty acids they are present in them, and they are good source of protein as well, right. This is called as a blue revolution. When we talk about the green revolution, that means overall increase in a productivity as per the agriculture is considered. Whereas the productivity, when it is considered with that of the fishes, this is called as a blue revolution. Now we know there are 2 types of species. So you have to explain about the blue revolution in detail. We have done the blue revolution in the sessions. So in this case, the 2 species, first of all we have to explain about the fresh water. In case of the fresh water, we have is a rohu and katla. Rohu and katla. Whereas for the marine, we have is a sardine and hilsa. Marine, we have is hilsa and sardines. This is what you have to explain in this. A blue revolution, try to explain the blue revolution in the detail. What are the different breeding methods that can be opted as per the blue revolution is considered. What are the different fishes also we have already discussed in the sessions. Now, let's move to another category of questions which is of a 5 marks. This is a 9th question. Read this question properly. By the time, let me prepare this board for you. State the objective of animal breeding List the importance and the limitations of the breeding. How can the limitation can be overcome? Give example of a new breed of each of the cattle and the poultry. Now, what is the objective of animal breeding? Animal breeding is what? This is a procedure by which a new varieties that can be obtained which are having a beneficial characteristics. What are the beneficial characteristics? We are talking about animal breeding. That means those characteristics which are important to us. So, basically the animal breeding means same. So, objective is to produce animals with desirable characteristics. With desirable characteristics. This is called as a animal breeding. So, in this case there can be a mating between a two type of individuals. What are these different type of individuals? These can be a mating between a superior female and superior male. You have to explain about both of these. What is a superior female? What is a superior male? You have to explain these things. Now, list the importance and the limitation of the breeding. In the question, in the previous question when I was discussing about that, we have discussed the major problem is that basically we have to discuss about the importance. First of all, let's discuss about the importance. Importance is that a good variety is obtained. A good variety is obtained which is having a desirable characteristics. Desirable characteristics. First of all, whatever the beneficial genes are, they get accumulated. So, beneficial genes get accumulated, and whatever the diseased genes are, diseased expressing organism, they will be removed by evaluation. Evaluation. Right, these are the certain importance. And also let's talk about some of the negative thing or the limitation. What are the limitations of these? Limitations, as I have discussed. First limitations are those, these are those in which the inbreeding depression comes. Inbreeding depression comes. And the second is that the overall productivity and fertility is reduced. Now, this question states how we can overcome such type of problem. This can be overcome by outcross. Even for a single generation, if we go for a outcross, whatever the problems which is associated with the breeding, inbreeding, that can be removed. Now, the answer, question number 3, which is of 9th third part, states, give an example of a new breed of each of the cattle and poultry. In the cattle, when I was discussing about the cattle, I have discussed about Hisar Dale. Hisar Dale, right, which is formed by mating between Bikaner ewes and Merino rams. Now, one more we have to discuss about the poultry as well. Poultry. In the poultry, we have an example of Hampshire. Hampshire. Hisar Dale and Hampshire, these are the two examples. This is for the cattle and this is for the poultry, right? So, now let's move on to the next question, question number 10. What is plant breeding? Let's two-step in a classical breeding, in which classical breeding involves. Now, let's discuss about steps. They are asking about the plant breeding. First basic procedure which we have discussed, that is, it is a prerequisite of a plant breeding, that is called as a collection of germplasm. Collection of germplasm. That means whatever the different alleles are there which is related to one genes, all are collected for a particular variety. After that is a selection of parents. Selection of parents is also very important, because if we want that such type of characters should be present in the progeny, we have to select these parents. So, with desirable characteristics, character, we have discussed about each of these in detail, and the topic says we have to explain only two, but let's discuss about some more points also. When you have to explain only 2 points, try to explain the 2 points in detail. Now, after that is a self-hybridization. Sorry, this is a cross-hybridization. Cross-hybridization. The next step is evaluation and testing of plants. Of plant. After that is testing and release of variety. Release of variety. Suitable variety, they will be released after particular certification. We have discussed about each of these points in detail, so you have to explain like this. How has the mutation breeding helped in improving crop varieties? Give an example where this technique has helped. Now we have to explain about the mutational breeding. Mutation breeding is that breeding by which the changes they were allowed to happen, and these changes which happened in them, that happened because of the mutagens. Mutagens can be any chemicals, they can be any radiation. Whenever they fall on it, changes in the nucleotide happen. We have discussed the same in detail in the last sessions, and in this case what we go for, we look for those varieties in which the changes has happened, a desirable changes has happened, that leads to the form. If some changes is there in the nucleotide, definitely there will be a change in the protein also. If there is a change in the protein, definitely protein will be having certain consequence. If suppose the changes which they are causing is resistance, if they are causing certain type of resistance in case of the plant, that will be selected. Now in this, let's talk about one examples. One example, you all must have heard of a mung bean, which is resistant to yellow mosaic virus, and powdery mildew. Right. So this is the example you can mention. How has the breeding program helped in improving the public nutritional health? State 2 examples in support of your answer. Now, in the sessions, hope you have noted down about the mung bean. Now, in the chapter when I was discussing in the sessions, we have discussed about the Atlas 66, which is used as a donor for the production of a high protein content in case of wheat. So in this you can take an example of a Atlas 66, which help in production of wheat, which is having a protein content, a high protein content. Right. Next is, we have already discussed about in case of corn. Corn are produced which are a rich source of certain amino acid, they have a high tryptophan content. And one more, high lysine content. These are the two essential amino acids. And this is how the biofortification is being done, and these are the two varieties which are obtained. This was obtained in around 2000. This is a fortified corn which is produced, and we have produced also the fortified rice as well, right? So students, we have discussed about around the 10 questions from a different category we have discussed. So hope the session is clear to you. We'll meet you next session. We'll discuss about a new chapter. Till then, take care of yourself. Thank you so much students for watching this.